So Lab 4 is designed to look at accessory structures to the digestive system. So our largest accessory organ is our liver, which is our second largest organ in our body. And the liver sits in the upper right quadrant of the abdominal cavity below the diaphragm. And the liver is separated into lobes by a ligament that's called the falciform ligament. And the falciform ligament separates the liver into its largest lobe, the right lobe. And the right lobe typically contains the gallbladder, which we can usually see toward the inferior part of the right lobe of the liver. And then the left lobe of the liver that we see over here. Now, once we take the liver out, we'll be able to look at a couple of other lobes of the liver. But if we take the liver out and we look behind the stomach, then we will find the other accessory organ, which is the pancreas. The pancreas sits in a loop of the duodenum, and we'll be able to see it better if we remove the transverse colon. So the pancreas sits in a loop of the duodenum. And the pancreas is larger in that loop, so we call that the head of the pancreas, and then the body of the pancreas. And then the pancreas tapers as it goes to your left side, back toward the spleen sitting right here. And so that's called the tail of the pancreas. And so let's go back now and look at the rest of the liver, and then we'll come back and talk about the detail within the pancreas. So last time we were looking at the liver from this view, where we could see the falciform ligament and the right and left lobes with the gallbladder down here, inferior on the right lobe. If we rotate the liver upward, we'll see that the falciform ligament actually separates and creates a ligament that runs across the top. Since this ligament sits under the heart, it's called the coronary ligament, and it's what helps suspend the liver from your diaphragm to keep your liver in place. Now, if we rotate the liver backwards and inferior so we can see it, then this is the back of the falciform ligament, and it's actually a ligament that is a remnant of your umbilical vein, which is the round ligament of the liver that we're seeing posterior. And if we look between the round ligament of the liver and the gallbladder, we see a rectangular mass of the liver. So this is the third lobe of the liver, which is called the quadratic lobe of the liver. So quad, four-sided, it's rectangular shaped. As we rotate the liver more posterior, then the large vein coming from the inferior aspect of our body, the vena cava, is located on the back of the lobe, on the back of the liver. And there's a lobe that sits on the left side of the vena cava, which is the caudate lobe of the liver. So here's caudate, quadrate, left lobe, right lobe of the liver from this posterior view. Now the liver itself is highly important to processing blood. So we have major blood vessels that come into the liver. So this large artery we see in the bottom of the liver is the hepatic artery. And then the hepatic artery is going to branch and go to different lobes of the liver. And then this kind of purplish blood vessel that we see here is a vein. And it's a vein that originated in capillary beds in the small and large intestine. So it's a vein where blood has been through one capillary bed before. So it was arteries to the small and large intestine, capillary bed, vein from those capillary beds to the liver, and then it's going to enter a second capillary bed in the liver. So when capillary beds are combined, then the blood vessels that supply them are called portal blood vessels. And so this is called the hepatic portal vein. And it's carrying all the nutrients that you've picked up in absorption through the small and large intestine. And so the liver is going to process those all for you. And then the liver, in the breakdown of red blood cells, produces a byproduct called bile that we use to aid in digestion. So all of the green tubes associated with the lower part of the liver are tubes that would be associated with transporting bile. And so what we can see if we look right here is that there are two branches right here that we can see. This one's coming from the right lobe of the liver, and this one's coming from the left lobe of the liver. So these would be called hepatic ducts, so the right hepatic duct and the left hepatic duct. Where those two ducts actually come together right here, there's a very short duct that we'd call the common hepatic duct. And we use the word common because two ducts are coming together to form that one. Now normally when you aren't dumping bile into your small intestine, then bile that's being formed is shunt through this duct, which is the cystic duct, to the gallbladder. And then your gallbladder gets larger as it stores more and more bile. And then if you ingest food, then there's a hormone called CCK that triggers the contraction of the gallbladder, and then that pushes the bile down the cystic duct again. And where the common hepatic duct and cystic duct come together, we have the common bile duct. And then the common bile duct is going to run inferior to the stomach down along the pancreas and dump into the small intestine. So we're going to look at that relationship next. So this model is good because it shows us the overall relationship of this duct system we were just talking about. So here's the right hepatic duct, here's the left hepatic duct. Where these two ducts come together is the common hepatic duct. Here's our cystic duct going to the gallbladder. There's a sphincter right here called the sphincter of Oddi. When it closes, then bile backs up. And so new bile being produced is going to come down the bile ducts to the common bile duct, and then being shunted into the cystic duct to fill in the gallbladder. When food enters the small intestine, then enteroendocrine cells in the crypts of Lieberkühn in the duodenum release CCK, and CCK gets its name because it causes contraction of the gallbladder. So cholecystol is a reference to the gallbladder. Cholecystokinin is active at the gallbladder. And so when cholecystokinin contracts the gallbladder, it also relaxes the sphincter of Oddi here, so that now bile runs down the common bile duct and dumps into the small intestine. And it helps emulsify fats and helps buffer the acidic chyme that was entering the small intestine. Now the pancreas is a gland that produces digestive enzymes, bicarbonate solution as a buffer, and then we have little tiny islands of cells in the pancreas called islets of Langerhans that are endocrine in function and produce hormones. So what we can see on this model is the part of the pancreas that's actually producing digestive enzymes and bicarbonate, which are called acinar or acinar cells or acini. And the bicarbonate and digestive enzymes run down the pancreatic duct to the duct of Weyersohn to this point. And then it branches, and we have a smaller duct that comes up here, which is the accessory duct or duct of Santorini, and it dumps pancreatic juice in here. And then the duct of Weyersohn joins the common bile duct that we just talked about. And the two then dump products together. So where the two ducts, the duct of Weyersohn and the common bile duct come together, we have an enlargement that's vase-shaped, so it's called an ampulla. So it's called the hepatopancreatic ampulla, or the ampulla vator. So now what we want to do is we want to look at the histology of the liver to understand how the liver actually works. So the liver is divided into these hexagonal structures that we're seeing here, which are lobules. So a pattern we're going to see this quarter in several organ systems is that we have an organ that can be divided into large subdivisions called lobes, and then what makes up lobes are smaller subdivisions called lobules. And so just always remember, a lobule is a small subset of a lobe. And so a lobule is constructed so that blood flows through it so that hepatocytes can actually process it. So a lobule is a six-sided structure. At each corner there are three vessels. A red one that's a branch of the hepatic artery, a purple one in this instance, which is a branch of the hepatic portal vein, and then a green one, which is a bile duct. So these three entities are called triads. And we actually have 6 triads associated with each lobule. At the center of a lobule, we have a vein that's going to collect the blood that's been processed. So it's called the central vein. And the way the liver works is blood is going to flow from these hepatic portal and hepatic artery through a ductwork system from the outside of the lobule toward the inside of the lobule, and then it's going to be processed. So if we want to look at that in detail, then what we're going to do is we're going to take part of one of these lobes and enlarge it over here. So we're looking at a lobule, and here we can clearly see the triad. And the way you can identify the subunits of the triad is that the bile duct is lined by simple cuboid epithelium. So we're going to see nice round nuclei. Both the artery and the vein are going to be aligned with simple squamous, but vein walls are very, very thin, and artery walls have smooth muscle in them. So this is a thicker wall and a much thinner wall. So under a microscope you can see that. So a branch of the hepatic artery, branch of the hepatic portal, and a bile duct. So what happens is blood from these two mixes. So it's one of the few places in our body where venule blood and arterial blood actually mix. And then they run through these canals that run through the liver, which are called sinusoids. So sinusoids are special capillary beds where the intercellular clefts are large enough so red blood cells can pass through them. And that allows the hepatocytes that line these sinusoids to destroy aging red blood cells and to modify nutrients that we have taken in. They can store carbohydrates in the form of glycogen. They can modify amino acids. They modify fats and create cholesterol and high density and low density lipoproteins, all for transport. So what eventually happens is the blood flows through these sinusoids and is processed, and then eventually ends up in the central vein, which has been cut here. And then we have phagocytic cells that we find wandering around in these sinusoids. So they look like the little starfishes that you're seeing on the little model. So those are called Kupffer cells, which is the short, easy name, or endoreticulite cells. And so what happens is the hepatocytes take the products of aging blood cells and dump it into a little tiny duct that carries bile. So it's called a bile canaliculi. And the bile canaliculi carries the bile to the bile duct, where then it eventually ends up in the gallbladder being stored for disposal. And so what we can see is the liver is a modified capillary bed that is critically important for modifying nutrients so they can be carried in the blood and utilized better by the body, detoxifying compounds that are toxic to our body, and destroying aging red blood cells. And then the end product of that is if it's something we cannot manage, we're going to put it in our bile and then get rid of it through feces.